here we go, and it says it right here. So in part one, we talked about kind of overall cell division, binary fission, but most importantly, we touched on a little bit of genetics, okay? We're going to spend a lot of time on genetics in Unit 4, but in order to understand cell division, you have to understand a little bit about why we have cell division, okay? We need to replicate those chromosomes and get them into each cell. So now we're going to talk about replication, or should I say just cell division in eukaryotic cells. We're going to go over mitosis. So I'm going to show you a video, an animation that actually has words to it. Okay, then I'm going to follow it up with my commentary, lucky you. But I'm going to end with another cool animation that doesn't have any words. And I want you guys to try and see if you can follow along and pick out what's going on when, okay? So let's start off with this lovely video, and then we'll come back together.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug-of-war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
La 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 la! Okay, so that was cell division in a nutshell. You're going to see lots of pictures that I'm going to show that look similar to some of these, okay? This is actually a slide of an onion root tip where it shows many different cells in different phases of cell division, okay? So you'll see some of these and hopefully understand it a little bit more as we go through. So let's talk about the cell cycle first, okay? The cell cycle is really important. This diagram actually tells a lot, okay? This entire circle is representing the cycle of the cell, okay? And if you notice, I'm going to point out some different things. This whole purple area, so basically from where I'm at right now, all around is actually interphase. But what goes on in interphase, which is the first kind of main cycle, and actually a majority of the time the cell basically stays in interphase. But in interphase you have cell growth, okay, overall growth of the cell. So basically after a cell has split, then you have cell growth, okay, so that cell remains in interphase and cell growth. Then you have DNA replication. Okay, you have to replicate that DNA because then it's going to basically be chopped in half so that it can be identical in the cells that it's going to be divided into, okay? And then the last part of interphase is preparation for mitosis. Spindle fibers form, different things prepare the cell in basically ready for all of these phases that we're going to talk about. So again, the cell cycle shows that the cell is actually in interphase most of the time, okay? But then we move on to actual mitosis. If you notice, just from here to here is mitosis, okay? And it covers these different phases that we're going to talk about: prophase, metaphase, anaphase, and telophase, okay? Cytokinesis is where the cell actually cleaves, it splits, okay? That is not necessarily a part of mitosis, so we don't really consider that part of mitosis. That's actually actual cell division, where that cell divides into two, okay? So it's still a part of, I should say, the cell division part right here, but it's not a part of mitosis, and it's not a part of interphase, okay? So you have interphase, prophase, metaphase, anaphase, telophase, and then cytokinesis, where the cell splits. So get to know that, understand it. It's a good timeline. It's a good thing to remember. You'll need to know it for your tests. All right. So let's move on to interphase. I showed you that basically the cell spends a lot of time in interphase, okay? So cell growth takes place, DNA is replicated along with organelles and other cellular components, and the cell prepares for cell division. Okay, so remember, this whole cell division is a constant cycle where you have a new cell, you go through interphase, then you go through this cell division again, go through interphase, so it's going to continue to go through this. So let's look at this picture again. As you can see, and you'll be doing a lab on mitosis, we're going to have to look at some different cells, and you'll notice that you can't really see anything specific going on here. Well, that's because basically interphase, things are just kind of growing, carrying on. You're not seeing the condensed chromosomes yet. So when you see a cell like this, pretty much you got interphase, okay? And you're going to see a lot of those because, again, most cells stay in interphase. Not most cells, all cells stay. That's the longest period of cell division. But let's move on to prophase. Prophase, the DNA recoils, the chromosomes condense, and the nuclear membrane disappears. Remember, you have a nuclear membrane that surrounds those chromosomes, okay? But actually in prophase, it starts to disappear because, again, we're going to have a lot of things happen with those chromosomes. So the mitotic spindles begin to form, and that's where you have those basically spindle fibers that will eventually pull these chromosomes apart. So again, let me read through that one more time so you get this, because you need to know these phases very well. DNA recoils, the chromosomes condense, the nuclear membrane disappears, and then mitotic spindles begin to form. Now you may not be able to see all of these things in these slides. Like the mitotic spindles, you may not be able to see those, but you can start to see that the chromosomes are condensing, okay? You can't see necessarily a pattern of them, but you can see that they're starting to condense, and you can start to see that the nuclear membrane disappears, okay? There's not this evident circle. I mean, you can see that it's in a circular form, but this membrane is starting to disappear. So there's prophase for you. Metaphase, kind of one of my favorites. My favorites because it's very easy to spot. So metaphase, the chromosomes line up along the middle of the cell with the help of those spindle fibers, okay? So they attach to those centromeres. And if you guys remember in part 1, I talked about the chromosomes, okay? You have the chromosome that kind of looks like an X. Well, in the middle, actually, each of those kind of X's are called a chromatid. And they are hooked together in the middle by a centromere, okay? So in metaphase, those chromosomes are going to line up along the middle of the cell, okay? So meta, kind of middle. And then basically those spindle fibers are going to attach to the centromere of each of those replicated chromosomes, okay? And I'll show you a good picture right here. So here's a plant cell, and here is an animal cell. You can see that here's a cell wall that only plant cells have, okay? And notice how they are all lined up right along the middle of the cell, okay? So metaphase is very easy to pick out, and you can really clearly see the spindle fibers here that are attaching to this condensed chromosomes, and you're going to see what happens next. But I like metaphase, very crystal clear in what's going on, okay? Anaphase. Now I don't want you to, I don't want you to get anaphase and telophase mixed together because there is a distinct difference, okay? So in anaphase, the chromosomes split in the middle, and those sister chromatids are actually pulled apart by the spindle fibers. And they go to the opposite ends, we call them poles of the cell, okay? So one's going to one side, and one is going to the other side. So you can start to see those pull away, okay? So let me show you that picture of the plant and animal cell. Here's again the plant cell. Very, very clear that those are being pulled to the opposite poles of the cell, okay? Same with the animal cell here. Now when I show you telophase, it's going to look very similar. But the big difference is there is no cell division starting yet, okay? We're still just dealing with kind of those chromosomes that they have now pulled apart, and you have those sister chromatids on either side, okay? So let's move to telophase. So telophase now, if you notice, the chromosomes along with the cytoplasm and its organelles, they start to divide into two portions. They haven't fully divided, because remember, that's cytokinesis, but they have started to cleave. So when I pull back and you guys can see these pictures, you can see this faint line here, okay? Now it hasn't divided fully, but it's starting to, okay? So that's telophase. So don't get anaphase and telophase confused, because it looks like, okay, yes, they're pulling apart in anaphase, but if you don't see that kind of separation of the cytoplasm, it's not in telophase yet, okay? So this is right before those two cells, or the one cell actually becomes two. So that is when we turn into Dun, dun, dun. Cytokinesis. So cytokinesis, the actual splitting of the daughter cells into two separate cells. So that's what we call cytokinesis. So you can see this picture here. This is where you're actually two cells. Now, really, cytokinesis, this little area right here would be like snipped. But pretty much you have two separate cells right here, okay? Where you're not still with two cells kind of conjoined and you just have this faint line like in telophase. You actually have kind of pulled apart, cleaved apart those two cells, the two daughter cells, okay? So then the cool thing is, then these cells are going to undergo interphase. So they're going to go through cell growth, they're going to go through DNA replication, and they're going to go through kind of preparation for mitosis, and then they will undergo that as well, and they will divide as well. So your cells are constantly dividing. It's very cool because that's how our bodies basically sustain themselves, because we are constantly dividing and making new cells while some of these old cells die off. Now there are certain cells that do not undergo reproduction. So one thing is our nerve cells. Once you have your nerve cells, that's all the nerve cells you have. They don't divide and we don't get new nerve cells. That's why if you have any nerve damage, you're done. You don't gain that back, okay? Unless there's still life in some of those cells. But if those cells have been cut off from nutrients and so forth, you're not going to regrow any of those. So very cool to kind of think about our bodies and when it comes to this, and that we are constantly making new cells and other cells are dying off. Now obviously as we get older and we get closer to our end of life, that's when basically we're not reproducing cells efficiently, or when we do reproduce them, maybe there's some glitches in them and we start to get diseases and sickness and cancer. So you guys will learn about cancer when you guys do your mitosis lab. It's something that I don't want to touch on a whole lot in here because I want you guys to get that from your lab, and we'll talk about that when we go over our lab together. So that is the cell cycle. Let's do a review here, and then I'm going to show you that last animation without words, which is kind of cool. I like the animation in this one, but see if you can follow along with what's going on. So let me pull this a little bit closer here, and we'll look at this. Okay, so again, this is a great little picture because it shows all of the cell cycles. So interphase, again, you have kind of cell growth, you have preparations for mitosis, and you have the DNA that's replicating inside this nuclear membrane. Remember, the whole thing is this is all happening in the nucleus, okay? Not within the cytoplasm. This is all in the nucleus. So you have an actual nuclear membrane that kind of houses that DNA, okay? So then in prophase, you have the microtubules that have already formed, okay? But now you have spindle fibers that are forming, okay? The nuclear membrane starts to disappear, and most importantly, those chromosomes start to condense. So you'll see them getting thicker, okay? If you look at them under a microscope, you'll see the thickness. All right, then, now we don't really talk a whole lot about pro-metaphase. I want you guys just to know metaphase. We could break this down a little bit further, but metaphase in general, those chromosomes line up along the middle, okay? We call that kind of that's why it's metaphase, middle, meta. So lines up along there, and those spindle fibers are basically connecting to the centromeres, okay? And in anaphase, those spindle fibers start to pull them apart. So you have sister chromatids, the daughter chromatids, that basically are being pulled to each side of the cell, each pull, okay? And then telophase is where you start to see that cleavage, okay? So it starts to basically divide, but cytokinesis is the actual division of it, okay? So there's a good little overview, but quick little review. Let's go into the lovely video, and that will be it for section 3. So kind of a cool little animation, makes you feel like you're like in the cell and you're a part of it. And it's kind of cool because at first it shows you in the cell, in the cytoplasm. And if you guys hopefully caught the mitochondria, they showed kind of a Golgi apparatus. And then they took you into the nucleus, okay? So I want you guys to rewind that, watch that again. It's a great video to really kind of see what's happening, and then you can start to remember what's happening in each of those phases, okay? So next time, we're starting section 4 will be meiosis and sexual reproduction. Lovely. Okay, until then.